quantitative trait loci. Before we do that, I want to give you just a little bit of historical background on how we originally began to understand and map genomes. So originally, all of this work, at least for eukaryotes, was done in the fruit fly. And fruit flies are useful for a number of reasons. They were easy to keep in the lab. They had these very large polytene chromosomes that occurred in the salivary glands, and so they were much easier to visualize than most chromosomes. And also, we were able to mutate them and find many, many mutants. And so the original way that we would actually map genes onto the genome is to do crossbreeding experiments with flies, and we would look for recombination. I'm not going to get into the details of that, of how that occurred here. There is a problem on worksheet number 3, which is posted now. You can look at it, you can begin working on it. I will post a separate video that will help you with worksheet 3. I know that we've done some of that in class, and in fact on most of the worksheets. If you've been in class, you followed along. But I'll post a little bit of a help for worksheet number 3, and of course you're always welcome to reach out to me and send me an email or ask during one of the Zoom sessions, which we'll have right before the exam. Worksheet 3, you still have a week and a half or so to do it, so just keep an eye on that. I'll remind you when it's due. So by using linkage disequilibrium and crossing over experiments, we could make a rough map of where genes were located. And so this is actually an early diagram of the Drosophila genome, A, B, C, D, and E, these different chromosomes, with different areas labeled and mapped through some staining techniques, and then different genes labeled. Okay, so the very first genome mapping was done with crossbreeding experiments. We have now modern methods. So one of the more recent methods is FISH. Now FISH is an acronym. It stands for fluorescent in situ hybridization. And basically it's just a way to light up different targets in a microscope image. So here we have two different genes, one that has been tagged with a red label and one that's been tagged with a green one. And so notice we can identify homologous chromosomes. So this may be chromosome number 1, and we've tagged two different genes on either end of these chromosomes. And so we know that these genes are linked to one another because they're on the same chromosome, but not very closely linked. They're on very, very different parts. So fluorescent in situ hybridization is a great way to visualize the actual location if you have a target, if you know what you're looking for. Today, of course, we do genome sequencing. And genome sequencing is getting the entire sequence of a genome. And we'll talk about that more in Unit 3.1c, where we start looking at genomes and genome sizes. Okay, but think of it as just reading off all of the at the molecular level, right? Adenine, guanine, cytosine, thymine, and reading that off and knowing the entire genome of an organism. But genome sequence is simply like taking photographs. So imagine you wanted a very detailed map of an area and all you had were photographs. It would give you some information, but it would be a lot nicer if we had features labeled on there, right? If you could say, oh, here's a city, or this represents a forest with a certain type of ecology. So in addition to just sequencing the genome, we would also like to map it, right? Show where the genes are, and then annotate it, make notes about what those genes do and how they interact. And that's a very, very involved, long process. And so although we've had a human genome now for nearly 2 decades, we're still in the process of mapping and annotating the human genome. It's very detailed now, but there's still a lot more work to do. And for most organisms, we're still in the early phases of sequencing and mapping, and in some way we've made more progress than others. Okay? So just realize that we can sequence entire genomes, but mapping them and identifying all the features on those genomes is another very long and engaging process. One of the very important and powerful tools that we have to do that is a QTL. Now QTL, which we have abbreviated here, is short for quantitative trait loci. Okay, and so it's kind of a long, complicated name, but at least it's fairly explanatory. A quantitative trait is any trait that you can measure. And loci refers to the location or the address on the chromosome. So with a QTL map, what we can do is basically identify neighborhoods in the genome of genes that contribute to an interest of trait. Okay? So imagine that we have 2 different strains of fruit flies and we breed them together, and then we get some sort of combination and we maybe could sort them out by loci. So this is a very tricky thing to do, and where maybe you can only look at, here they're looking at 3 different genes and maybe trying to figure out if they were linked to one another. They could do crossbreeding experiments, but that's just for 3 genes. The QTL is a way to look at the entire genome at once, and here's how it works, okay? So I'm actually going to fill this in as we go, and you can write notes if you want, write it down. It's always helpful to write things down, even if you don't look at them later. Sometimes just the act of writing things down will help you, okay? So let's put these in here as we go. All right, I'm going to make this a little bit bigger. Okay, so number 1, to do a QTL study, the very first thing that we need to do is have a sequenced genome. Okay, so let's do that. 1, sequence the genome. That's maybe a gimme, but we'll put it here anyway, okay? The next thing we have to do is we have to identify genetic markers. And these can be their number of genetic markers. The most common one is called a SNP. You don't need to remember that name, but a SNP is an acronym that stands for single nucleotide polymorphism. And basically what it means is if we look across the entire population at that one particular location, right, at that locus, that some individuals have one nucleotide, like a thymine, for instance, and other individuals might have a guanine. So there's variability there, right? It's a polymorphism, okay? Once we identify those genetic markers, then we need a large data set. Now, when I say data set, what I'm talking about is some sort of genetic database. So maybe 1,000 or 100,000 individuals that we've got their genetic markers sequenced. So we know what genetic markers they have, and then we know a little bit about their medical background. Okay? So for instance, let's say we were interested in heart disease. Is heart disease phenotypic in origin or genotypic in origin? Now, think about it for a bit, and hopefully you've answered both, right? Heart disease is genetic, it can be passed down, but there's also a large environmental role. So it's maybe 60-40, 50-50, somewhere in that neighborhood. So there certainly are genetic features, and we would want to understand those genetic features so we could study them, okay? And so what we would do is we look across this large data set, and we would find everybody that was susceptible to heart disease. And we maybe even break it down by types of heart disease, but we find everybody who has that condition, we put them in one group. And then we take everybody else, the general population, they'd be in the other group. And then what we would do is we would do this large analysis. So we would analyze for correlation. And what I mean by that is we would look to see if across all of these genetic markers, let's make just a little bit of space here, across all of these genetic markers, if there was any correlation, right? So we've sequenced the genome, we've identified all these genetic markers, we've got this big, large data set, and then we've analyzed them for correlation. So basically we'd say, hey, at genetic mark number 1, which is at the beginning of chromosome number 1, do people with heart disease have more of a chance of having a T at that position than the rest of the population? And most of the time the answer is going to be no, it's the same as the rest of the population. So we move on, genetic marker number 2. Is there a correlation between this genetic marker and heart disease? And we do that for every genetic marker all across the entire genome. And what we find is we will find a number of genetic markers, maybe like right here, an area where the genetic markers in that area have a skewed. So maybe it should be 60-40 for that genetic marker. But in the heart disease population, we notice that it's 80-20 instead. So that would be a significant difference. And we would see that in this map. So this would be the beginning, like chromosome number 1 at the end and chromosome number 2 at the end. So we see a strong area in this location for a correlation. Now, that does not mean that that specific genetic marker causes heart disease. All it means is that it's in the general area. It's in the neighborhood. And so now we have a focused area of the genome. Rather than trying to individually test every single gene we identify across this chromosome, we can say, oh, here's one for heart disease. Now, the cool thing about this is if you've got a big data set with lots of background information, you could look for heart disease. You could look for Alzheimer's disease. Does that have a genetic component? Yeah, some, right? So you could look for it. Could you look for like eye color? Now, that may be low on the priority list because it doesn't have a huge medical impact, right? But schizophrenia, type 2 diabetes, any sort of disease that has a genetic component, the QTL study with a big enough data set could be used to identify and map and annotate genes that contributed to that. So this is the way, and we're continuing to use it, but this is the way that we have found many, many different loci, and then been able, location, and then be able to identify specific genes within that location that contribute to a specific genetic condition. And there's actually, I'm going to open up a web browser here. There's actually a very interesting and large data set. There's some privacy issues that are involved here also. But in Iceland, there's this massive genetic database that has been created by the government. And this is done, again, maybe you have some privacy issues with this. I think I might a little bit, but it's mostly anonymous. They won't tell you who it is. It might just say, a 35-year-old woman, and then with a medical background that's there. So you can actually, this is an interesting article. You can just Google Iceland genetics and you'll get this one, but it'll give you a talk about this, and there's tons of papers that have been written on it. Many of the things we know about the human genome have come from this large database where basically every citizen of Iceland has their DNA sequenced for these genetic markers, and enough of a medical background that many of these traits can be correlated with specific regions. So it's a little interesting read. It's not real long. If you want to take a look at this, it's kind of for popular interest. It's kind of cool. And this is becoming better and better that we're getting bigger and bigger data sets. We do need to balance it a little bit with privacy issues, but it's an incredibly powerful tool that will allow us to do all sorts of things with these large databases. And so QTLs will look like this. We have high correlations in two areas. We identify two loci on this chromosome that we would want to look for in that area for some causative effort. Okay, and so now QTLs, in addition to FISH, like I was telling you about, and just doing the plain old sequencing and old-fashioned crossbreeding experiments, we can add a QTL as an incredibly powerful tool for identifying any genetic condition. And we're focusing on diseases. That's where the money goes first, but we really could use any genetic condition, and we could identify regions of the genome that contribute to that genetic condition. So that's a QTL. Study it, review it. Again, on worksheet 3, you'll get a little bit of an exercise looking at crossbreeding experiments and how we map genomes originally.